Hello everyone. Today we're going to talk about neurons and the cells that support them that are known as glial cells. Let's talk about the structure of a typical neuron. A neuron is basically a nerve cell. Neurons can actually live for a pretty long time. Our neurons can last us for over 100 years. Most neurons cannot regenerate, by the way. So if they are damaged, they're not going to be able to regenerate themselves. There are a few exceptions, especially the neurons that help us make memories. The three parts of a neuron are the following: a cell body, an axon, and a bunch of axon terminals. Cell bodies, right here, if we have a bunch of cell bodies in our central nervous system, which is basically our brain and our spinal cord, a cluster of those cell bodies, we call it nuclei. If you have a bunch of cell bodies in your peripheral nervous system, which is basically consists of like your cranial nerves and your spinal nerves, a cluster of cell bodies in your peripheral nervous system, we call it ganglia. Inside of the cell body, there are organelles, such as nucleus, mitochondria, ribosomes. Inside of the cell body, proteins are being made and chemicals known as neurotransmitters. So neurotransmitters and proteins are made within the cell body. Once neurotransmitters are made, the neurotransmitters are going to be sent down the axon and they are going to be stored within the axon terminals. Now, there are those spiked looking like regions attached to the cell body, but it doesn't always have to be sticking out of the cell body. Those dendrites, those are known as dendrites. Those dendrites are basically our receptive regions. They are responsible for receiving signals, let's say from other neurons, and then they take those messages, they take the messages that they've just received and they send those messages down to the cell body. Now those messages that are being sent from the dendrites to the cell body, those are basically electrical messages, or we refer to them as nerve impulses. The nerve impulse that's being sent from the dendrite down to the cell body, we refer to it as a graded potential. The part right in between the cell body and the axon, this region right here, is known as the axon hillock. Now, the graded potential, as I said, is a message that runs from the dendrite to the cell body. For that message to go down the axon, that message will have to pass down the axon hillock. And once it passes down the axon hillock, and it's going to go down the axon, we don't call it graded potential anymore. We call it action potential. So an action potential is the electrical message or the nerve impulse that runs down the axon. Now, if you look at the axon, you will notice that the axon is covered up by this insulator layer right here, known as the myelin sheath. So the myelin sheath acts as an insulator. It insulates and it protects an axon. Not all axons of our neurons are myelinated. Another reason what's the function of having a myelin sheath other than insulation? It allows the message to run down your axon faster. So without a myelin sheath, that nerve impulse, that action potential, is going to go down the axon. It's just going to take a longer time. There are three shapes for our neurons. There are unipolar neurons, bipolar neurons, and multipolar neurons. A unipolar neuron looks something like this. So here's the cell body. This whole entire thing is actually the axon, and here are the dendrites. Unipolar neurons, most of our sensory neurons actually look like unipolar neurons. We're going to talk about what sensory neurons are in just a few. Bipolar neurons, they look something like that. Bipolar neurons are pretty rare. We don't have a whole lot of neurons that look like that. The ones that look like that are the ones found in your eyes, in your ears. Another example of where you would find bipolar neurons are neurons in your olfactory mucosa. Multipolar are the most common, so most of the neurons in your body look like that. They look like multipolar neurons. And most of your motor neurons also look like that. Sensory neurons are the neurons that are going to pick up the message and send the message to your central nervous system. So basically to your brain and your spinal cord. Motor neurons are going to pick up the message from your central nervous system, and it's going to send it away from your central nervous system to your effectors. So let's say that something was like itching you. So you want to go ahead and you want to scratch your neck, right? So the reason why you are feeling that your neck is itching you in the first place, well, because you have some skin receptors, and the message is going to go down your sensory neuron to your brain and your spinal cord. And your brain and your spinal cord, they're going to receive that message, they're going to process it, and they're going to tell you what's the best thing to do. And how's your brain and your spinal cord going to send that message? Well, they're going to send the message down a motor neuron. That motor neuron in this scenario right here is going to take the message to your effectors. The effectors in this case would be your skeletal muscles in your arm and in your hands so that you can reach out to your neck and you would start scratching it. So those effectors don't always have to be muscles, by the way. They could be muscles, they could be glands, depending on the scenario that you are talking about. Now, if you take a look right here, you would see that most sensory neurons, they do look like unipolar. Same shape right here as the unipolar neuron. And most motor neurons, shape-wise, they are multipolar. Now, there is a neuron that you can find between a sensory neuron and a motor neuron, and that neuron right here is known as interneuron. Inter means in between. So an interneuron is a neuron in between a sensory and a motor, and it just helps take the message from the sensory, kind of send it to your motor neuron. If you look at this part right here of the spinal cord, you would notice that the spinal cord has two colors. There's this region right here, which we refer to it as the white matter. And the reason why it's whitish in color, because it's mostly made out of, or it has a bundle of myelinated axons. This region right here, we refer to it as the gray matter. And the gray matter, this is where you have a bundle of those clusters of cell bodies. And remember how we said clusters of cell bodies in your central nervous system? We refer to them as nuclei. These, by the way, are nerves. Nerves are a bundle of axons. So this right here is one nerve. If you open that one nerve, you're not just going to find one axon, you're going to find a bundle of them. Let's talk about the cells that protect or support our neurons. And those cells are known as glial cells or we can refer to them as neuroglial cells. Now, we have some glial cells in our central nervous system, so embedded in our brain and our spinal cord, and we have ones in our peripheral nervous system. Let's start with the ones embedded in your brain and in your spinal cord. We have 4 of them. So let's talk about the first one, known as our astrocytes. Our astrocytes look something like this. Astro actually means star, and cyte means cells. So astrocytes, they are like your star cells. And astrocytes, as you can see, they are clinging on to your neuron. So here's your neuron right there. And at the same time, they're grabbing on to close by capillaries. And some of their functions, why we have astrocytes, is that they help support and brace your neurons, kind of anchor them in place, and they help anchor them to a nutrient supply line. So they're going to help actually by grabbing onto the capillary, they're going to be able to pass on some nutrients to the neurons. So they can help exchange materials between the capillaries and the neurons, like nutrients. Astrocytes are also known to control the chemical environment around the neurons. So if there are extra ions, let's say potassium ions found around your neurons, your astrocytes can actually help mop them up. And your astrocytes can also help like recapture ions found around the neurons, and they can help recycle them. They can also help recycle released neurotransmitters released by your neurons. Astrocytes, they also help create what's known as the blood-brain barrier. So think about astrocytes. When they cling on to those capillaries, they can actually kind of like cover up those capillaries and create a barrier. That barrier is important because your capillaries, which are basically your blood vessels, they carry nutrients, but they also carry waste. And so you don't want the waste to get out of your capillaries and enter into your brain tissue. So your astrocytes basically prevent any leakage or movement of any unwanted substances from your blood into your brain tissue. Another type of glial cells we have in our central nervous system are cells known as ependymal cells. And our ependymal cells, they look something like this. They have a bunch of cilia, those hair-like structures on their surface. And having cilia is going to allow those ependymal cells to actually, their cilia is going to start swaying back and forth, and they can start swaying back and forth the cerebral spinal fluid. So they can actually help move that cerebral spinal fluid. Ependymal cells, you would find them lining the hollow cavities in your brain, which we refer to as our ventricles. Ependymal cells help create a barrier actually between your cerebral spinal fluid and between the brain tissue or the spinal cord. Another cell we have are cells known as microglial cells. Microglial cells, they're known to be phagocytes. What are phagocytes? Phagocytes are cells that eat up any foreign particle. So the microglial cells, if there are foreign particles, and not just foreign particles, if there are dead particles in the brain tissue or in the spinal cord, the microglial cells are actually going to move up to those particles and they're going to go ahead and swallow them up and break them down. So the microglial cells do not allow any basically debris to be found in your brain or in your spinal cord tissue. They will clean it up right away. Another cell is known as the oligodendrocyte. This is how the oligodendrocytes look like. And if you notice, the oligodendrocytes, they send out these projections and they start actually releasing the myelin sheath. So the oligodendrocyte is the one responsible for making that myelin sheath. And as you can see, it deposits the myelin sheath around the axon. Now, and they can actually myelinate several axons at the same time. Notice how it doesn't myelinate the whole entire axon. Notice how there are gaps in between the myelin sheath. Those gaps are known as the nodes of Ranvier. So these right here are known as the nodes of Ranvier, which are basically gaps in between the myelin that was just deposited on the axon. The glial cells found in our peripheral nervous system, we have two of them. We've got satellite cells and we've got neurolemocytes. Our satellite cells, they are usually in our peripheral nervous system surrounding the cell bodies. And if you remember, we said that if we have a bunch of cell bodies in our peripheral nervous system, we refer to it as the ganglion or ganglia. Now, those satellite cells surrounding the cell bodies, their function is actually very similar to an astrocyte. So they kind of do the same thing as astrocytes. Neurolemocytes, there is actually another name for neurolemocyte, Schwann cells. So you can either call them Schwann cells or neurolemocytes. And your Schwann cells, they function as the oligodendrocyte. So the Schwann cells are basically responsible in making the myelin sheath and myelinating the axons of the neurons in the peripheral nervous system. And same thing when they go ahead and they deposit that myelin sheath around the axon, there are going to be gaps known as the nodes of Ranvier. This image right here shows how we have multiple neurons connected to each other. So let's say that you want to send a message from your central nervous system all the way to your big toe in your foot. Is it only going to take one neuron to send that message? Nope, it usually takes several neurons. As you can see right here, the neurons do not touch each other. So there are usually gaps in between the neurons, and those gaps, we refer to those gaps as the synaptic clefts. So let's go ahead and take a look at what's going on right here. So here is the neuron with its cell body, its axon, and the axon terminals. Now, if we zoom in on the axon terminal, so let's go ahead and zoom in on the axon terminal. What you would find inside of an axon terminal, you would find a bunch of what we call the synaptic vesicles. Inside of those synaptic vesicles, that's where the neurotransmitter is stored. Which part of the neuron is the receptive region? The dendrites. So these are representing the spikes, right? The dendrites. So once those dendrites pick up a message, the dendrites are going to send an electrical message or a nerve impulse down to the cell body, represented by those orange arrows. So those orange arrows are representing the graded potential, that nerve impulse that's moving from the dendrites to the cell body. The purple arrows are representing the action potential. So the action potential is the nerve impulse that's going to run down the axon until it reaches the axon terminals. Now the thing is that once the action potential runs down the axon and reaches the axon terminals, that's going to trigger those synaptic vesicles to open up and release the neurotransmitters. Now, where are the neurotransmitters going to be released? The neurotransmitters are going to be released right here in the space between the two neurons, and that space is known as the synaptic cleft. Once the neurotransmitters are released in the synaptic cleft, what's going to happen is that those neurotransmitters are going to bind to the dendrites of the neuron that's found right here, and another electrical impulse is going to be triggered, and the electrical impulse, one more time, that graded potential, is going to run to the cell body. Then another action potential is going to be triggered. The action potential runs down the axon until it reaches the axon terminals, and then same thing happens. Those vesicles are going to release the neurotransmitters in the synaptic cleft. The neurotransmitters bind to the dendrites, a region of the neuron, and another graded potential. And as you can see, the cycle is going to keep repeating until you hit the final destination. So let's say the message needs to go to your big toe. Until we hit that muscle of your big toe, that cycle is going to keep moving from one neuron to another neuron. Now, the neurons found before the synaptic cleft, we refer to these neurons as the presynaptic neurons. Pre meaning before. Or you can think of it in another way. Instead of thinking of the presynaptic neurons as the neurons before that synaptic cleft, you can think of them as the neurons sending the message towards the synaptic cleft. The postsynaptic neuron, you can think of it in two ways. Post meaning after, so they are after that synaptic cleft, or you can think of them as the ones taking the message away from the synaptic cleft. But the thing is, if you have more than one neuron, notice how this neuron, it's a postsynaptic neuron to the neuron before it, but it's actually a presynaptic neuron to the one after it. So the terms presynaptic and postsynaptic, you can actually have a neuron that's both. The connection between your neurons, by the way, we refer to it as synapse. So you have synapse between several neurons. This image right here explains why when an action potential goes down the axon, goes down into that axon terminal, why do the vesicles release the neurotransmitters they're carrying? So normally inside of your neuron, the condition inside of your neuron, it's negative compared to the condition outside. So inside our neurons, the condition is more negative. The electrical state is more negative compared to outside. The thing is, as an action potential runs down the axon and reaches the axon terminals, the inside of your neurons, or in this case, the inside of your axon terminals, it becomes more positive. So the electrical state changed from being negative to positive. What's going to happen is that when it becomes more positive now, a voltage-gated calcium channel is going to be triggered to open. And calcium is actually going to start rushing inside of the axon terminal. Once calcium is rushed inside of the axon terminal, the calcium is going to bind to some special proteins inside of your axon terminals, and the vesicles are going to be triggered to release the neurotransmitter that they are carrying. Once the neurotransmitter is released in the synaptic cleft, the neurotransmitter is usually going to bind to some special receptors found on the surface of the postsynaptic neuron or the postsynaptic membrane. And we're going to talk about it in more detail later, but at least that introduces to you what happens when an action potential runs down the axon terminal. The inside becomes more positive. That triggers a voltage-gated calcium channel to open. Calcium is going to start rushing inwards, causing the vesicles to open up and releasing the neurotransmitters in the synaptic cleft.